Bonsoir and welcome to this fifth episode of the Late Night Conference. My name is Wilhelm Huck and I'm your host tonight. I really appreciate it that you're with us tonight again, because I know that slowly bars and restaurants are opening up and there might be other competing interesting things to do on a Thursday evening. But rest assured, we're already thinking about how we are going to make sure that also next season you will have a very entertaining show with us. You remember last month Professor Cronin showed us some very intricate mass spectrometry and computational methods to try and quantify whether, well, even complex molecules could be biological or abiological. And today we are going to take a little bit deeper look into how such molecules could be made. And in biology, there are networks of enzymatic reactions. And you could think about the Krebs cycle or the Kelvin cycle that convert a series of substrates very elegantly and precisely into the building blocks that every cell needs. However, on prebiotic Earth, those enzymes and their cycles, they didn't exist. Or maybe they did. Maybe the cycles were already present on the early life, and life only invented the enzymes to run them more efficiently. And well, if that is really the case, then the presence of such cycles will be really important in an alternative look at the origins of life. And you remember Professor Shostak talking about the RNA hypothesis and the RNA world. But of course, you could also imagine that instead of making RNA, you would first make all the small molecules. And this would be the theory that says, well, there was first a metabolism, and all the reaction networks existed, and they produced all the necessary building blocks. And our guest tonight is Professor Joe Moran of the University of Strasbourg, who is interested in exploring the type of chemical reaction networks that could have been formed on very early Earth. And very specifically, he's looking at those kind of networks that could have acted as prebiotic equivalents of the citric acid cycle and related cycles. To give you a little bit of background, Professor Moran studied chemistry at the University of Ottawa, and he also obtained his PhD there. And after postdoctoral work at the University of Texas at Austin, he took up an assistant professor position in Strasbourg in 2012 at the ISIS, and I will not say that in French because every time it's a bit of a tongue twister, unfortunately. It's the Institute of Supramolecular Chemistry and Engineering, where he has been ever since, rising through the ranks very rapidly. He's now the director of the Laboratory of Chemical Catalysis and full professor. His group is working on prebiotic chemistry, but also on catalysis and metal organic, bioorganic chemistry, and has expanded very rapidly. He has been supported by an ERC starting grant and very recently also an ERC consolidator grant. Before we start, let me remind you that we are looking for interesting questions from you, the audience. Put them in the chat and we'll pick them up, and I will then convey them to Professor Moran, and we'll have a discussion after his talk. And remember, the questions can be about anything, about the science that he is presenting, but also maybe on the sort of more general aspects of how you start a career in the academic world. Also, please make sure you like the video. Remember to subscribe to our channel. Click the bell so you will get notified for our next episodes. And with that, we are ready to start. Joe, the Zoom is yours.
Okay, thank you. Big thanks to you, Wilhelm, and to your team for organizing this wonderful late night conference series. I've really enjoyed watching the other episodes, and I consider it a privilege to be here. So I'll do my best to explain, as you said, why I think that the first step in the origin of life was the onset of a self-organized chemistry that may have been not so different from metabolism. Oh, all right, so before I do that, I just want to say that we all stand on the shoulders of giants. And because this talk is only 25 minutes long, I just want to acknowledge some of the people whose ideas, experiments, and even criticisms really have shaped my view of the origin of life. And in particular, I want to acknowledge Harold Morowitz, Bill Martin, and Eric Smith. And you can also check out our review on experimental work on non-enzymatic metabolic reactions listed below if you like. All right, so there's no question that the origin of life is a multifaceted problem. And you've heard on this conference series or elsewhere that the origin of life is a problem of information, or it's a problem of self-replication, or chemical synthesis, or compartmentalization. And I think all those things are true, but I would argue that the underlying principle that supports all of those things is that life is a problem of chemical self-organization. So, we know that chemistry had to self-organize towards complexity on the path to life, but the question is how and why would this have happened? And a chemist usually increases the complexity of a molecule by guiding it through a series of specific chemical steps. So each under specific conditions in a specific order, adding reagents just at the right time. And this is extremely unlikely to have happened in a continuous way on a lifeless planet, which is a problem because things that are highly unlikely to have happened almost by definition are not good explanations for things. But despite that, a lot of chemical research on the origins of life still works within that paradigm. So how can we explain the spontaneous emergence of complexity? And I think we have to rely on chemical self-organization from the very beginning. So every system we know of in science where dynamic order forms spontaneously works in essentially the same way. They're called dissipative structures, and they form because they're the paths of the paths of least resistance to releasing pent-up energy in the environment. And they only form when a specific type of energetic stress acts on specific types of building blocks. And even then, only under a specific range of conditions. So below you can see one example, a hurricane. So a hurricane only forms when a specific temperature gradient occurs in the ocean, must be over a specific distance scale, in this case, thousands of kilometers. But if we want dynamic chemistry that self-organizes on its own, becomes more complex on its own, it can't happen in the way a chemist normally does synthesis. So it's not really self-organization. So we have to instead look for some sort of chemical hurricane. And by that, I mean a network of chemical reactions that forms spontaneously as the chemical path of least resistance in order to release up pent-up energy in its environment. So the products of one reaction will form the starting materials for the next one without anyone intervening in the experiment. And this sort of thing probably, it's only going to happen under very specific conditions, just the right types of molecules, the right types of energy, the right temperature, the right catalyst, pH, and maybe even with oscillations or gradients of those things within a particular region of space. Now, what makes self-organized systems robust is that they exhibit self-reinforcing feedback that keeps them going. And in chemistry, the self-reinforcing feedback mechanism is called autocatalysis. We say that a single chemical reaction is autocatalytic if one of the products is a catalyst for the reaction that produced it. So, in this case, B. But those are relatively rare, at least I think. But it's also possible to achieve autocatalysis in a network of reactions, even if none of those reactions is catalyzed per se. And this is often called network autocatalysis. So imagine a molecule A that reacts with some reagents in the environment to give B. It goes on, reacts with more reagents to give C, and then to D. But then D splits up into two to give back one molecule of A and one molecule of B. That means each turn of that cycle doubles the number of molecules that it contains. And this sort of exponential growth could concentrate these reagents found in the environment into the intermediates of the network. Now, of course, we all know that there will be side reactions, there always is, desired or otherwise. So if this network isn't going to die out, then material will have to flow into it faster than it flows out. And here the network can get some help from reactions that pump additional matter into the cycle, stabilizing the overall network. So a bit later in my talk, I'm going to show that the core reaction cycle of biochemistry has exactly this property of network autocatalysis, and it can be stabilized by another very simple biochemical pathway. And I'll try to make the case that those reactions could have given rise to life. Now, before I do that, there's a few things I'd like to discuss. The first thing is, how does a complex reaction network emerge in the first place? Can we just mix a whole bunch of random reactive molecules together and see what happens? I think the history of this science shows that when you do that, you usually end up with tar. But we have learned how self-organized complex systems emerge in other fields. And it generally happens when a bunch of simple rules work together. And a classic example is the computer game called Conway's Game of Life. So in this game, simple time-dependent rules act on black and white pixels, and most combinations of rules produce a mundane result, in fact. But some combinations of rules result in these complex lifelike patterns. And these rules are, of course, set by the programmer. But if we want self-organized chemistry that can eventually work with evolution, the rules, or more specifically, the chemical mechanisms, need to be dictated by something outside of a molecule's intrinsic reactivity in order that the rates and selectivity of those reactions can change in the future. And I think that means we'll need naturally occurring catalysts or an ensemble of catalysts that can promote a number of simple mechanisms at the same time. Now, I think that core metabolism looks like it could have emerged this way because it uses about 7 reaction mechanisms over and over again. All right, so if we're going to do chemical synthesis in a true network, this inevitably creates a tightly interconnected web of reactions, and that naturally leads to hierarchical organization within that network. This is true of any synthetic network in principle, but I'll use how biochemistry works today as an example. So matter flows in as geochemicals, such as CO2 and hydrogen, ammonia, phosphate, metals, etc. And these make metabolites. First, it makes carboxylic acids, and some of these are keto acids. And then these make amino acids and sugars and lipids. And then amino acids and sugars can react to make ribonucleotides. All of these things can react further on their own with each other, or they can polymerize to make functional polymers like proteins, or genetic polymers like RNA and DNA. But that web of interconnected reactions that emerges in this or any self-organized synthesis network starts to get really hard to change because it contains compounds and reactions that play many different roles at once. So it's kind of like trying to take a card out from the middle of a card tower. Well, as far as we know, the core network of biochemistry hasn't ever fundamentally changed throughout the whole history of life. And that's despite 3.5 billion years of evolution acting on it. So I'd argue that for the same reasons, it probably hasn't drastically changed since prebiotic chemistry either. So what I'm saying is that evolution has never really been completely free to innovate. The complex interdependencies that support evolution and allow it to operate in the first place also constrain what it can change. And which is why I and a number of other researchers in the original life field think that that prebiotic chemistry would actually have a lot of similarities with the core of metabolism. Now, we've only talked about chemical synthesis so far, but if we really want to think of life's emergence as a dissipative structure, I think we also have to consider breakdown. And in that sense, life is a lot like a fountain, right? The fountain's pump burns fuel, which throws water up in the air, and the force of gravity pulls it back down. And what we call a fountain is actually not really a thing, but it's more of a dynamic steady state. So the water molecules are in constant turnover, but the dynamic pattern of the fountain persists. And life's chemistry kind of does the same thing. So redox agents, which are mostly reducing agents, working with phosphorylating agents, drive chemistry towards complexity, and then oxidizing agents and hydrolysis break it back down. What we call life isn't really a thing either. It's a dynamic steady state pattern. The molecules are in constant turnover, but the dynamic pattern persists. So for synthetic chemists like myself, this means we'll have to change our typical mindset. We're usually geared only towards synthesis, and obviously that's important, but breakdown is also an intrinsic part of life. Okay, so just to recap quick, the chemistry that led to life must have been self-organized from the beginning, and I argued that the thermodynamic driving force that allowed it to persist was probably the same type that brought it into existence, meaning it probably would have been driven by redox reactions and phosphorylating agents. The need for self-reinforcing feedback means autocatalysis would probably have been important. And I argued that a complex reaction network will only emerge when a small number of reaction mechanisms can operate together in a controlled way. So this means that naturally occurring catalysts are likely to be involved from the beginning as well. And then when you get a highly interconnected web of reactions, they become hard to disrupt. So prebiotic chemistry could still bear some resemblance to biological metabolism. Okay, so notice that with the exception of the point in red here, none of those predictions actually required knowing anything about how life works. They're just consequences of general ideas about non-equilibrium systems and complex systems. Okay, so everything I mentioned so far is really just a prediction, nothing more. So it's meant to focus our experiments in productive directions, but I think it implies that we should be able to reproduce the chemistry of core metabolism, or something close to it at least, in the lab without enzymes using naturally occurring catalysts, and then infer from that the energy source and the environment that produced it. So here you see a crude roadmap to get us started. It's a minimal set of metabolic pathways that would get us from CO2 to genetic molecules, namely RNA. And the flow of matter starts with the pathways that convert CO2 to life's universal building blocks. And I'll talk more about that in a minute. But that's the acetyl-CoA pathway and the RTCA cycle. The RTCA cycle has the property of network autocatalysis. And when it's combined with the acetyl-CoA pathway, which feeds into it, we get the stabilized network autocatalysis that I mentioned earlier. So if we have that, then we'll need something like gluconeogenesis to make sugars. We'll need amino acid biosynthesis to make probably 4 or 5 amino acids. And finally, we'll need ribonucleotide biosynthesis, which comes from sugars and those amino acids. And this is, this is really a big, probably decades-long experimental challenge. And until 7 years ago, only a handful of reactions in this network were known to occur non-enzymatically, let alone whole pathways. Okay, so now I'd like to tell you a little bit about our experiments. So we first began our work with life's CO2 fixing pathways, and surprisingly, at least to me, despite 4 billion, or 3.5 at least, billion years of evolution, there's only 6 different pathways that life uses to fix CO2, to make the molecules that support metabolism. And there's only one that doesn't build onto already existing biomolecules, and that's the acetyl-CoA pathway, also known as the Wood-Lungdahl pathway. And the pathway is found in both bacteria and archaea, which are the 2 major branches of the tree of life. And a lot of evolutionary biologists think that this pathway was already present in life at the time of its last universal common ancestor, or LUCA. So on a chemical level, what this pathway is doing, it's reducing 1 molecule of CO2 to carbon monoxide, reducing a second molecule of CO2 transiently to something equivalent to a formal group, and then on to a methyl group, which is bound to its transition metal cofactor. And then there's a migratory insertion event to form a new carbon-carbon bond, and that gives us an acetyl group. And finally, there's a reductive carboxylation to give us pyruvate. Now, this is biology I'm talking about, but I'm using the language of transition metal catalysis here because essentially every mechanism of this pathway involves transition metals, either in the cofactor or in the enzyme itself. So the question my team was wondering was, could this pathway have operated with metals alone before enzymes altogether? And the short answer, we think, is yes. So we noticed that the 0 valent forms of the metals used by the cofactors and enzymes of the acetyl-CoA pathway, they react with CO2 in warm water to produce formate, a methanol, and acetate, which is a C2 compound, on the near millimolar scale. So in the case of metallic iron, nickel, and cobalt, pyruvate is also produced, which is a C3 compound in the acetyl-CoA pathway. And so to explore this further, we decided to focus on metallic iron, since it makes up 80% of the core of the Earth, and it's arguably the most important metal for biochemistry. And we found there that the reaction works over a broad range of temperature and pressure, as low as 30 degrees Celsius and as low as atmospheric pressure of CO2. In fact, at 1 atmosphere of CO2 and 30 degrees Celsius, the only organic products we detected are acetate and pyruvate. So in other words, the same metals used by the biological acetyl-CoA pathway without any enzymes fix CO2. They produce the same C2 and C3 molecules of the biological pathway and essentially no other organics. However, the organisms that run the acetyl-CoA pathway don't use metallic iron as the source of electrons to fix CO2. They use hydrogen gas. And these tend to be organisms that live in hydrothermal vents where hydrogen gas is abundant. So we teamed up with the team with Bill Martin, who's a professor of evolutionary biology in Dusseldorf, and with Harun Tusut, who's an expert in heterogeneous catalysis at the Max Planck Institute in Mülheim. And we found that several iron-based minerals that are present in hydrothermal vents, such as magnetite, which is Fe3O4, greigite, which is an iron sulfur mineral, and awaruite, which is an iron nickel alloy. They're all able to promote hydrogenation of CO2, giving the same products I showed in the previous slide. And the minerals are catalysts. They don't change during the reaction as determined by Hurun's team XRD analysis. So this is an even closer non-enzymatic mimic of the acetyl-CoA pathway. Okay, so now I'm going to switch gears. I'm going to talk about a second CO2 fixation pathway called the RTCA cycle. It's also known as the reverse Krebs cycle. And this one's important for two big reasons. The first is that it contains all 5 of the universal synthetic precursors to biochemistry. So all lipids are ultimately made from acetyl-CoA. All sugars either come from pyruvate or oxaloacetate. All amino acids come either from pyruvate, oxaloacetate, or alpha-ketoglutarate. And some cofactors come from succinate. So the important point is that with those 5 molecules, we can build up all of biochemistry. They're all present on the same cycle, and it's the only cycle in biochemistry that has that feature. Another way of saying that is that biochemistry is what you can make from the RTCA cycle. Now, the second reason this cycle is important is that it's autocatalytic, or network autocatalytic, and I already explained why that's important. It starts off with 1 molecule of acetyl-CoA. It builds until it gets to oxaloacetate. It continues to build until it gets to citrate, which is 6 carbons, and then that splits into 2, giving back 1 molecule of acetyl-CoA and 1 molecule of oxaloacetate. So each turn of the cycle doubles the number of molecules contained within, but that exponential growth in this case is even more interesting because it happens to concentrate geochemicals into the critical building blocks for life. Now, 5 of those reactions, of the 11 reactions in that cycle, require ATP. The others do not. So the question is, for us, again, could this cycle or something similar to it have operated before enzymes as prebiotic chemistry? So there had been some earlier attempts to create a non-enzymatic RTCA cycle. I don't have time to go through all of them. It was done by George Cody, Scott Martin, Marcelo Guzman. But the difficulties encountered in all of those cases was the ability to string together successive reactions. So our approach was to divide the RTCA cycle into 2 sequences. And to make our lives easier to start, we examined only the ATP independent steps. So the first sequence we looked at involved the reduction of oxaloacetate to malate, its reversible dehydration to fumarate, and its reduction to succinate. And the second sequence involved the reduction of oxalosuccinate to isocitrate, its reversible dehydration to aconitate, and its reversible rehydration, but now on a different carbon, to citrate. So to search for conditions where those reactions could occur spontaneously without enzymes, we screened for temperature, pH, reducing agents, or simple ones anyway, and metal ions as catalysts. And here's what we found. So many different reducing agents did the job for those different reduction reactions, but our friend metallic iron, once again, interestingly, did it quite well. And zinc, we found, was the best ion for promoting the 2 dehydrations, and chromium the best one for promoting the hydration back to citrate. So a lot of those reactions work at room temperature if micelles are present. Without micelles, most of them work at 70 degrees Celsius, and all 6 of them work if we go to extremely hot and acidic conditions. So those are almost certainly too harsh to be relevant to the origin of life, but I consider the work as a proof of principle that all 6 of the ATP independent reactions can work non-enzymatically. They can even work together under just one set of conditions without enzymes, just using simple metals and metal ions. Okay, so after that, we put a fair amount of effort into trying to get the ATP dependent reactions of the RTCA cycle to work without enzymes. But we just hit a wall there. In fact, we didn't really know it at the time, but what we were doing was we were stuck in a local minima in our search. And sometimes you just have, it's the way it is in science, you just have to put things aside for a while, try again with fresh minds and fresh hands. And so last fall, we went back to it. And it turns out you can make some of the ATP-dependent reactions occur after all, and under exceptionally mild conditions, in fact. And those conditions also happen to make all of the ATP-independent reactions I mentioned on the last slide work as well. So now we have a catalyst that can do 7 consecutive reactions of the RTCA cycle at room temperature in neutral water. So we're still making headway there, but as you can imagine, these folks are now furiously working to see if we can complete the entire cycle. Okay, so in parallel to those efforts to uncover a non-enzymatic RTCA cycle, we wondered if there was a way for similar chemistry to happen without ATP or a simpler analog thereof. And so we got inspired by an analysis done by Daniel Segre's team together with Hyman Hartman, which asked whether metabolism may not have originally started off with all of the elements we know today, so carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, but whether some may have joined later in stages. And so they concluded that a large part of core metabolism could still function without phosphorus. And this is the network that they described. It still contains all 5 of the universal precursors to metabolism I mentioned before. They're circled in black. And of the compounds that are made up only of carbon, hydrogen, and oxygen, like those in the Krebs cycle, the 2 best connected were pyruvate, which I just told you we could make from CO2 using hydrogen and iron, and glyoxylate, which is circled in red here. It's a very reactive aldehyde, C2 compound. So this map suggests that there could be parts of a proto-metabolic network that could still function without ATP or some simpler phosphorylating agent, and that it could have relied very heavily on those 2 compounds. And we already knew that those 2 compounds react in an aldol reaction due to the work of Springsteen and Krishnamurti, even without a catalyst at all, to form a new CC bond. And so naturally, we wondered what metal catalysis might do to such a system. So we found that when you mix pyruvate and glyoxylate in the presence of ferrous iron at 70 degrees Celsius, a network of reactions occurs. It generates a variety of compounds in addition to CO2. And this plot shows the 16 compounds that we were able to track over time. The network that we found involves 9 of the 11 intermediates of the RTCA cycle, and we can directly detect 4 of the 5 universal precursors to biochemistry that I mentioned earlier. The 5th seems implied by the reactivity of the network. So we focused on ferrous iron because it's widely thought to have been a major component of the oceans of the Archean eon. But in fact, similar reactivity we observed for other divalent metal ions, so manganese 2, zinc 2, cobalt 2, nickel 2, copper 2, and probably many other ions work as well. We didn't check exhaustively. But acid alone, or a monovalent salt like potassium chloride, for instance, do not catalyze the reaction. So we tried to piece together a mechanism for this reaction network, which is really not trivial for something this complex, as Wilhelm knows. The key parts are an aldol condensation between pyruvate and glyoxylate, followed by a reduction to give alpha ketoglutarate, and then another aldol reaction with glyoxylate. And after that, there's a series of dehydrations and oxidative decarboxylations, both of the products of that and the other intermediates I mentioned, that lead to the compounds of the RTCA cycle and other compounds as well. So the important bit here, I don't want to get too much into the organic chemistry, but the important bit is that these are the same types of reactions one finds in the RTCA and TCA cycle. In fact, several of these are the same reactions found in the TCA cycle, which is the oxidative version of the cycle that runs in humans. So reductive and oxidative reactions are happening in the same pot. Synthesis and breakdown are happening in the same pot. All of the chemistry I talked about so far happens without any human intervention. And this shows us that a complex non-enzymatic reaction network that has some similarities to metabolism to both build and break down at the same time is possible. Now, just for fun, if we do make an intervention in the experiment and we add hydroxylamine and iron metal to the mixture at a specific time, the keto acids are converted into the corresponding amino acids. So pyruvate gets converted into alanine, glyoxylate into glycine, ketoglutarate into glutamic acid, and oxaloacetate into aspartic acid. Now, hydroxylamine probably isn't compatible with the chemistry of the larger network, but it's still neat, I think, that these transformations are quite similar to how amino acids are made in metabolism. Okay, so we're not the only lab that does this sort of thing. So Marcus Ralzer's lab in the UK and now in Germany reported that exposing sugar phosphates to iron-rich warm water caused them to decompose by a chemical path resembling glycolysis, which is the main path that life uses to break down sugars back into pyruvate. And he also found that this happens for another pathway known as the pentose phosphate pathway. And this chemistry can even produce ribose 5-phosphate, which is one of the key intermediates in the biosynthesis of RNA. Now, of course, one has to have the sugar phosphates already to break them down. So my lab's actively working on a non-enzymatic gluconeogenesis, which is the path that life uses to build sugars up. Okay, so that brings me to the end. I hope I was able to convince you why it's important to know whether the chemistry of core biological pathways can happen without enzymes. A lot of the ideas I presented here have been around for a long time. They're not from me, but none of the experimental work that I've shown you in this talk is more than 7 years old, and the work from my lab is no more than 4 years old. So we're still actively working to see whether other core metabolic pathways can happen without enzymes as well. And I hope the next 5 years should show or allow us to have a more informed discussion about what role metabolic pathways may have played in the origin of life. So just before I end, Wilhelm asked me to say a little bit about my career. I guess a lot of this is a bit redundant with what he said in the introduction, but I'll do it anyway. So I was born in Montreal, Canada, and I grew up in a nearby suburb, and that's where I learned how to speak French, which I didn't realize at the time, but it ended up being really important for my career. Otherwise, I probably wouldn't live in France. And I wasn't much interested in science in high school when I was growing up. I was actually more into theater and music, but I ended up going into chemistry as a path to get into medicine, which is what my parents had suggested I should do. But after a couple of years, I got to do research in the summers and in both organometallic chemistry and catalysis and organic chemistry. And I realized that research was probably the thing for me more than medicine. So then I decided to do a PhD, as Wilhelm already said, in organic chemistry. I became the first PhD student for a young assistant professor, and that was a great experience for starting my own lab later. I worked on synthetic methodology, photochemistry, organic catalysis, all of which are probably, are peripherally at least related to the origin of life, if not intimately related. And then I spent 6 months at the National Research Council of Canada, working with John Pizzacchi, and there I learned a little bit of chemical biology, which maybe changed my perspective a little bit too. Then I moved to Austin, Texas. I worked for 2 years in the lab of Mike Crochet doing transition metal catalysis and asymmetric catalysis. And then I came, as Wilhelm said, to ISIS in Strasbourg, started my own group, and yeah, I've been here ever since. So if you have any questions about the career end of things, please ask in the question period. Yeah, so last, I just got to thank my group. So I'm truly grateful to all the members of our group, past and present, to our collaborators. I showed their names already on the relevant slides and their photos. Thanks to the ERC, Marie Curie Actions, and the Volkswagen Foundation for paying the bills. And finally, thanks again, Wilhelm, and your team for inviting me to be part of this fantastic late night conference series. And I'm happy to take questions.
Thank you. That was a fantastic presentation. Thank you so much. Really insightful and, well, in vein with the theme, coming full circle, where you showed the chemistry that you wanted to do and then the networks that followed. Really very impressive work. Thank you. I'm sure there will be questions, and they're coming in. I can see them on my screen. Just to recap, I think you made an excellent case for the importance of self-organization in chemical reaction networks as being at the very heart of what living systems must be able to possess in order to become alive. And I think you also showed that if you really want to have a self-sustained, self-organized system, then you need feedback in there, you need autocatalysis in there, and you need reactions that feed into that system and also molecules that are spun out at the other end. And then I think in the second half of your talk you showed that you don't need the enzymes, you can use the metals either as, well, metal zero or metal ions as catalytically active species, and although they probably lack the selectivity that you will find in enzymes, they clearly were able to carry out all, well, if not all, most of the steps at least in your biochemical inspired transformations. So very interesting indeed. I had one question that maybe we can come back to later on, but do you think that life could exist without the reverse TCA cycle or the TCA cycle? Because you said it's so preserved over the last 3.5 billion years. All trees of life possess that cycle. It almost begs the question that if you didn't have that, life wouldn't be possible. Would you go along with that suggestion?
It's really hard to say, but I mean, you've probably had this discussion in other contexts where when asked if you take into account that you need to form bonds between atoms, and there's only certain types of atoms that are going to be good at that, you probably need to exchange gases in order to allow a lot of kind of a biosphere to emerge. And so then you have to wonder, okay, well, what gases besides CO2 can allow this sort of thing to happen? I mean, HCN might be possible, but it's not clear if that is viable or not. So there's not a lot. I don't think there's a lot of options really. If you look at silicon, you go down go down a little bit in the periodic table. Okay, that has it can form bonds too, but it's not usually a gas. And yeah, it likes oxygen perhaps a bit too much and ends up being silicate all the time. So I think if you kind of consider all of those little details here and there about what is a viable, what are viable feedstocks for any self-organized chemistry that's going to happen on a planetary scale, then I think it limits your options quite a bit. And so if that's the case, then maybe the TCA cycle is one of only a few solutions or the only solution to these problems. It's just speculation, but it's completely possible, I think. Now,
yeah, that's no, go ahead. Sorry.
Yeah, just to say, so I think I've kind of viewed the origin of life as like a series of bottlenecks, and you have to kind of pass. And so it's possible that self-organized chemistry can happen with other elements under different conditions. But the question is, will they be able to pass all those bottlenecks that ends up producing something that resembles life as we would describe it? And I don't know, but I suspect it might be actually quite limited.
Maybe this is also a good intro to the next question that I see on my screen from a person called Black Skimmer, who says it's easy to find a hurricane dynamic structure in nature. Why not a chemical one? Where are the wild BZ reactions? So I guess if I translate that, it ties into the discussion that we just had, where you pinpointed the TCA cycle as one of those key dynamic structures in a chemical network. What could be the other ones? Why don't we have many more to play with?
Yeah, I think that's okay. I think that's it links perfectly with what I said in the last question. So there are we know oscillating reactions. There's whole communities of chemists working on those types of things. There's many reactions that exhibit complex behavior, but the question is, can they form bonds that can lead to increased complexity? And the BZ reaction doesn't do that. So I think so once you start adding each of those constraints that you would have to have and kind of like line them up and multiply them by each other, you're not left with so many options. So I think that is probably what makes the TCA cycle so special, is that not only does it have all those properties, but it makes everything from the environment, simple gases, but it happens to make synthetically relevant materials that are the starting points for all biosynthetic pathways, and not every cycle will do that.
Yeah, I was thinking about that, because it will obviously produce those molecules, but it will produce quite a lot of different molecules. And you could say that in a living system, it's very much constrained, and it actually does produce those molecules that life needs. But I'm sure in your experiments, you showed very nicely that you made lots of these compounds, but you probably made many more as well. If anything, you made them in low concentration, so they might become very difficult to analyze, which means that in a non-biological setting, those networks exist, well, almost like as a plurality of multiple cycles. How do you see some kind of evolution taking place there that shaved off all these other pathways in such a way that it became a more pure cycle?
Yeah, so this is something that's been discussed a little bit. So in 2015, there was a paper in Scientific Reports, Zubrev et al. So Alana Sparuguzik, the corresponding author, and they asked that very question. They used graph grammar models to create reaction network trees that could predict what would happen, and they found their conclusion was that you would get to much more complex cycles than the RTCA cycle, and the RTCA cycle is basically the shortest loop redundant version of that larger network. And I think that's a good approach.
If I remember correctly, it also said that in that same paper that the TCA cycle was not even the most plausible one from a thermodynamic point of view, if I remember correctly.
Right. And so I'm not, I think one of the things that's missing in that study is that there might be a lot more mechanistic constraints on the chemistry than what were seen there. So for instance, if a metal ion is catalyzing these reactions, that will select certain pathways out of the many ones that seem plausible on paper. It can make only a few of them actually possible. And so I think we have to take that into account. Just because it's thermodynamically possible doesn't mean it's actually going to happen. And I wouldn't be surprised, at least what I've seen in the lab, on that project, is that there's actually, you actually don't go off the rails as much as you might think you might on paper.
Okay, thank you.
Yeah.
This maybe is also a good point to go to the next question. Daiki Tanaka asks whether pyruvate and glyoxylate be made in the same location and set up the same reactions in the Krebs cycle under the same conditions.
Could they? Yeah, you mean if you took pyruvate and glyoxylate and tried to kind of merge the RTCA cycle with the thing we did in that other paper? Yeah, and so we are looking at it a little bit. I think it might be possible, but can they be made in the same location? I don't know. So there's been a paper recently. So when we published our paper, we made references to some older work that had shown electrochemically you could make glyoxylate from CO2, and we had done it with pyruvate, and so had others. There's recently been a new work that came out that's mostly theoretical, but it also proposes that you can get, using copper catalysts, glyoxylate fairly selectively. Whether that bears out in the lab, I don't know. Actually, we haven't looked at that much. We did it early on, but we haven't gone back to it recently. So I do think it might be possible that you can do both at the same time. That would be a lot simpler than trying to find the CO2 fixing steps that we are looking for. But I don't know. The other thing is we also look beyond the TCA cycle and look at the other reactions that involve phosphorylation, like in gluconeogenesis. And the motif that keeps on happening that's always required is phosphorylation. So the glyoxylate reaction doesn't require phosphate at all, but the CO2 fixing ones do. So that's why we're still not ruling them out, and we're going back and looking at them once again.
Thank you. The next question is from Daan van der Zwaag. Hi Dan, good to have you on the show tonight. Dan says, very interesting lecture, Professor Moran. You convincingly show chemical conversions and catalysis, and briefly mention the effect of micelles. How important is the spatial distribution of input and output?
Yeah, that's a great question that we don't really have any answer to either. I mean, in those particular reactions, what was important? So we tried many micelles, and in fact, we tried, I think, around 50. Only one worked. It's the one that's in that paper. It was one from Bruce Lipschitz's lab, and it's not prebiotic at all. It's just it's man-made. But the unique property of that micelle was that it makes nanoscale micelles, whereas the other ones are much larger typically. So the effect that we see there may not be so much, well, I guess it is a compartmentalization effect, but it may be the fact that you're kind of on a very small scale concentrating molecules, and you get these sort of small scale effects that you won't get in larger micelles. So I don't know if it's just compartmentalization that's important, or other aspects of chemistry that were critical. But in general, I don't know. I mean, if you look at the autopoiesis model that it's often talked about from Tibor Ganti, and he suggests that you need compartmentalization right from the beginning. And in fact, the steps that lead to fatty acid synthesis are very simple too. They're just Claisen condensations and dehydrations and reductions, the same type of reactions we use, well, except for the Claisen condensation, which is not that different. They're the same types of reactions we use in the rest of metabolism. So it's completely conceivable that those could happen abiotically as well. We've only investigated that a little bit and didn't see anything, but of course we are always changing our perspective as we discover new conditions, and we have to revisit that in the future.
Thank you. Then Enrico Orsi has a question who says, very cool presentation. In biological systems, enzymes catalyze reactions with substrate concentrations in the micromolar range. How difficult is it to drive the reaction with non-enzymatic catalysis?
I guess we don't know. So Marcus Ralzer's work is done on a much lower concentration regime than ours. That's more probably realistic. in biology. The issue with us, the reason we haven't done that yet is because our analytical capabilities are limited by what we actually had in the lab. So, I'm a synthetic organic chemist. I had a GCMS in my lab. I didn't have, GC, triple quad or whatever that Marcus has. So now we have a LCMS UPLC QTOF, which is much more sensitive, and we can start to do things on lower concentrations, but it wasn't possible before. So we will see, I suppose.
Yeah, I guess also there's in principle no reason why lower concentrations wouldn't work, except that it might take longer to get to a higher conversion. But I think it's not limiting the actual conversion, I suppose.
Yeah, it's a good, what that links to though is an idea we thought about at the end of our review when we wrote that ChemRev. Could it also be that the concentrations, the relative concentrations of metabolites are also limited by self-organization? And the concentration regime that life has chosen is a result of that initial self-organization to some extent. Because if you look at, it doesn't matter what organism you look at, the concentration regime of metabolites is always in the same range. And it varies greatly from one to the other, but they always tend to be around the same. So there might be something there too that is linked to self-organization that hasn't been appreciated.
Thank you. I think, Will, there was a question from Hector Soria from Spain, but that is gone now. But so I will ask a question from, there it is again. Hector Soria says, good evening from Spain. I was thinking if you ever thought of mixing peptides with metal ions in the Krebs cycle, it should mimic metalloproteins. And thanks again for an inspiring talk.
Oh, thanks. Yeah, so we have thought of it. Have we done it yet? We were supposed to. It was supposed to be a collaboration as part of an ITN network that got a little bit delayed due to COVID, but we will get to it eventually.
Very good. I don't know if Hector Soria is your collaborator there on that project.
No, I don't think so.
Maybe he should be, yeah. So.
Yeah.
I think we're getting slow. I'm just looking. No, we have some more time for questions. I thought I saw a question from Martin Feitus popping up on interesting to see biochemistry starting off without phosphorus. What about sulfur, which features strongly in Wachtelshaus's iron-sulfur world hypothesis? And I think I know that you have a paper on that on Archive, at least, where you are working on sulfur.
Yeah, we did. I think that's one of the amazing thing about research is you can take a lot of different turns and then you can turn back. And so after we saw that, for the same reason that we did the work I showed you that was in Nature, that paper suggested that thioesters could be replace phosphorus chemistry, or not replace, but at least operate without phosphorus chemistry. And so we decided to look at what happens with thioesters. And we did that. We found a way to do the oxidative decarboxylations in such a way that they were trapped as thioesters. And we showed that you could kind of mix the two things together. You could take the pyruvate and the glyoxylate and you could make a similar network and then trap the keto acids as thioesters in one pot. It didn't work super well, and it required some oxidants that were a bit extreme, I would say. But we also found UV light can do it. But in the end, what happened is the thioesters just sit there. They're like rocks, and they don't do anything interesting. And so that led to another question, which someone else in my lab is addressing. We have, in France, you have a CNRS, which allows permanent researchers to work in your lab. So one of the coworkers in my lab who's trying to start an independent project is trying to address that question. Why are the cofactor structures the way they are? Is it just a coincidence, or do they have some function that is not yet understood and in a non-enzymatic sense? And,
that's a very interesting line too.
That's for her to look at. I'm not moving into that area myself.
Connie Meinert has a question. She says, hi. Did you run the iron 2 promoted synthesis and breakdown as a closed system? What would happen if you extend the reaction beyond 48 hours and if you run it in an open system? Yes,
we're going to do that. That's another thing we were supposed to do and just never got around to it. But yeah, so we were going to get syringe pumps and do that. And we did get the syringe pumps, but we haven't done it yet. Just to see exactly what happens in a steady state. Because the reaction kind of goes to equilibrium after about 14 hours or whatever it is. So if we could just pump things in at the right rate and vary the rate, and then we would know. We're also, on that related note, we have within that same ITN, we have some modeling, kinetic modeling of with the data that we have so far, which is not great, but you can still start to model on it. And that might help us predict exactly what rates we should need to add those things in to get interesting behavior. I don't know whether it'll be predictive or not, but that's that was the idea. So we've done that, and it's almost complete.
Okay, I would like to encourage the audience, if they want to ask non-scientific questions and not iron 2 related questions, that is perfectly fine too, by the way. So that's just an encouragement for people to ask all kinds of questions. In the meantime, there is one not on iron 2 but on magnesium. Dan Viering says, thank you for the great lecture. Given the fact that magnesium 2+ is a cofactor for hundreds of enzymes, have you also looked at the potential of this enzyme for this metal ion for non-enzymatic catalysis?
We have, and we do find it does a lot of interesting things. Not so much in the Krebs cycle, but in other pathways, and that's to come.
Okay.
But yes, it definitely does. Yeah.
That sounds good. There is a question from Louise who says, how has COVID affected your research at Strasbourg?
It hasn't been as bad as it could be. So we were shut down like many other people from May to July roughly, or May to end of June. I'm sorry, March to end of June last year. So that affected our research because when we got, not only did we lose the time, but some people's postdocs expired during that period. They had to go back, and we had to get new people in. And all of our instruments seemed to break down when we got back because they hadn't been used in such a long time. Like the GCMS and various things needed to be kind of worked on. And then because of the lockdown, a lot of the companies that sell those instruments, they had service contracts to do. They couldn't come in quickly, so it took a long time for us to get back up and running with our key instruments. And so now I'm trying to double down and have double duplicates of all these things in case one breaks. We have still one that works, and I'm trying to we'll see how that works.
I could also add to that answer to Louise. I could say, well, in many ways, COVID has affected us negatively, but I think one of the good things is that it has taught us that actually having these kind of conversations that we have tonight are being made possible via remote video conferencing, which we probably would never have done. We would have waited till we had an occasion to share a conversation at a bar or something, but then nobody else would have been able to listen. So this is also a very positive thing to come out of it, I think.
I agree.
Let's see. I think we're slowly moving towards the end of our conversation. One of the questions that we try to ask every month is that every scientist has individuals who are important to their career. And you already mentioned that you had been at several places in several people's lab, where you mentioned that you did your PhD with a professor who was just starting as an assistant professor. Are there other people who you say, well, have really helped me, and how did you develop that professional relationship?
Well, every PI I worked with during my training helped me in a different way. John Pizzacchi helped me to learn a bit of perspective on biology that I didn't have at all. Working with, as an undergraduate with Susanna Scott, that was my first foray into research, and she really put me in a lot of amazing situations. If I have 2 minutes to tell you, I can tell you a funny story. I mean, it happened after 9/11, so this is how old I am. I was 2002, and 9/11 had happened a little while ago. And the lab that I was working in had a lot of postdocs from Pakistan and Afghanistan and Iraq, and she was moving from from Canada to the United States, and I think there was some difficulties getting a lot of her coworkers to shift over. So even though I was an undergrad and only worked in her lab for 4 months, she allowed me to go over to the to UCSB and to work in the synchrotron in Stanford, and I got an amazing experience. It was the first time I'd ever been on a plane in my life, and I got that great experience just by sheer chance and of having to be not from those countries, I guess, and that was an incredible experience that she had that faith in me to do that. And then Mike Crochet, taught me how to run a big group and different ways of doing that, and writing papers efficiently and that sort of thing. And he's given me lots of support over the years too. So everyone, and then here, the senior PIs of ISIS, particularly Jean-Marie Len and Jean-Pierre Sauvage and Thomas Ebbison, who I collaborate with, have been all critical in their own ways. So I think those are the major ones. Yeah,
Yeah.
and everyone was different. So
Sounds very, very important.
yeah.
Another important bit is asked by Stefano Mazzanti, who says, "Thank you for the amazing talk. What would you suggest to young researchers to find their own research topic?"
Oh yeah, that's a really good question. I mean, for me, it's always been to try to find something that is a little bit outside the box of what's normally done in your field. So, if you ask, when I talk to other synthetic organic chemists who come from the same, we did our postdocs together, they're like, how did you get into these crazy fields? How did you get into origin of life? Why would you do that? I just went to talks that were not on my topic, and which happens often in ISIS, for instance. And I went to one on origin of life. Like the first week I was an assistant professor here. It was given by an astronomer, and I don't think that I pulled anything specific out of that talk, but just got me thinking about origin of life. And I hadn't, it's a topic I thought was too obscure to work on, at least coming from North America. And then I came over here and was like, oh, that seems to be an acceptable thing to do. So I started thinking about that. I had a lot of crazy ideas which look nothing like what I do now. And I just had an open mind so I could look at all these different proposals and ideas and pick the one I thought made the most sense to me and identified a place that had not been worked on, which was the experimental part of this metabolism first idea. Lots of great ideas, not a lot of experiments. And so I thought, hey, I can do experiments. And
that's definitely true what you say there. So that's definitely a good advice. I think pick an area that seems important and that has not been very well developed so far. So well done to that. I have a very important person on the show tonight. It's a person called Jane Moran, who says, Professor Moran's toddlers are shouting, "Hello, Dad." So
wonderful. So, hello, Freddie and George.
Freddie and George, hello. Hello, Jane. Nice to see that you're on the show.
Wonderful.
I think with that, we're going to close for tonight. I also saw David Meringer saying that he tunes in every month. I think he said he found another great experience and an astonishing lecture. An inspiring lecture from Professor Moran. Thank you very much. He also thanks me for once again an astonishing late night conference. I don't think that is the case, but thank you all for tuning in, and thank you, Joe, for giving us a wonderful and inspiring presentation tonight. There were lots of questions, and I'm sure we could talk on for a long time. It would be good to share a beer now, but you have to go back to your toddlers. And it's getting dark behind us as well. So thank you again. Thank you, and we'll hopefully meet again shortly. Before we end, I also want to thank all the people on the team here for making it happening tonight once again very smoothly. Very nice. Everyone out there, please stay tuned for one more episode next month with astronomy topic in that case. In the meantime, watch back the video, like the video, and sign up for our channel, and we hope to see you next month. Thank you all, and goodbye.